thank you, Stuart, for the nice encounter we had here and also for the introduction. Well, you've heard from Chris about the phytoproducts of hemp, and I want to talk a little bit about the biology of hemp. So, we know from way back that hemp and its products, cannabis, have a lot of biological properties. Okay. How are we doing now?
A little bit better.
Okay. Okay. So, and it's been used pharmaceutically and therapeutically for many years, way back from China, Middle East, etc. And about 50 or 60 years ago, there's been an effort to develop medications based on this. And actually, there's been quite successful approaches. Pfizer had a big program, lasted about 12 years. Eli Lilly, in fact, Eli Lilly came up with a drug out of there. However, it didn't really go very far. The only drug that came out of a big effort was the Lilly drug, Nabilon, and I'll talk a little bit about it. And one of the problems, in addition, of course, was the sociological problem, which you heard here, when anything that had to do with this was bad, etc. However, there was a real problem because we didn't know how this thing works. We had no idea about the biology of hemp. It was thought that it works through membrane perturbations, etc. And until about 25 plus years ago. But at that time, the National Institutes of Health decided they want to go a little deeper into this. And they asked me to help them organize a conference, which happened in Bethesda, Maryland. And we invited all the people who were involved in cannabinoid science. And it was a very productive meeting because 2 years later, it turned out that the discovery of the endocannabinoid system happened, which is around late 1980s, early 1990s, and I was very happy to be part of this this process. So we know now quite a bit about the endocannabinoid system. We don't know everything, so I want to tell you a little bit about it in this presentation here. But let me just give you a little bit of a tidbit, because by discovering the endocannabinoid system, we come to find out this is a biochemical system that is very wide, very large. Keep in mind, for example, the cannabinoid receptors that are in the brain are the most numerous receptors in the human body. If you look at the brain, you look at dopamine, acetylcholine, norepinephrine, serotonin, they all are lower densities of the cannabinoid receptors. It's a very major system. What are the effects of cannabis? Well, as you may know, the red in the eye, decrease intraocular pressure, and it has been used for people who have glaucoma. And there are some good efforts happening as we speak to develop medications for glaucoma. Sense of dryness in the mouth, sensation of heat or cold, a big effect in the heart, which starts out early on and then fades away. And then there is a relaxation in the muscles. So what are the constituents of cannabis? We have, and you'll hear more about this from Josh Hassel, who's been following my presentation. We have the so-called cannabinoids. Cannabinoids are a special group of chemicals. They fall under the designation of cannabinoid terpenes. There are other terpenes, flavonoids, hydrocarbons, amino acids, etc. A very wide mixture. And in some ways, the effect of this, the biological effect of this plant is due to this combined effect of all of these constituents. We don't know all the details about it. So this is THC, which is the main constituent of cannabis. And you notice that it does have this oxygen here, hydroxy group. It's a phenol. That's why this thing turns brown as soon as you isolate it. It gets oxidized, and it's a tricyclic compound, and it has a tail. That tail is important because we find out that the longer you increase the tail, the more potent it becomes. There's a whole history there, and a lot of work on this. So And here are the constituents. So we have minus delta-9 tetrahydrocannabinol, which was discovered by Meshulam way after a lot of the British and American chemists started synthesizing these, but he was able to isolate this and synthesize it. And you see all the other constituents that are all related. And if you look at these lines here, you see there's some correlation here. For example, look at this one here, tetrahydrocannabinol. Then we take this ring and we make it aromatic, it becomes cannabinol. You can do that chemically and enzymatically. Or you could take this double bond and move it from one place to another place here. It becomes delta-8 THC, similar to delta-9, it's more stable. Or you could take this molecule right here. And then think about, see this bond here? If you break this bond here, then it gives you 2 hydroxy groups. And guess what this is? Cannabidiol. So there's a very good relationship between THC and cannabidiol. Is this little bond. You break this bond, it goes from here to here, cannabidiol. Now, to get all of these reactions going, as I said, we'll hear more about them later, these things have to, there's different pieces and we turn around and cyclize. And one of the products, before we get to the cannabinoids, is cannabigerol and cannabichromate. So from therapeutic point of view, there are really 3 drugs that are out there. We all know about Marinol, which is given in little capsules, in oil, and I hear that it's not a very effective drug. It doesn't sell a lot, etc. There's been a new product that came out of a British company called GW. What they did is they mixed 50-50, and this is, by the way, a botanical product. It's not a synthetic product. And they called it Sativex. It is administered in the mouth, so they have a little piece of equipment there. They spray it in the cheek, and it's unclear exactly how this goes in. One of the claims of the company is that it goes straight to the brain. However, there's more evidence that this thing gets absorbed and you swallow it, and it's essentially it has similar properties as Marinol. And this is a question of debate here. I'll be happy to talk about it if you have any questions on that. Now, this other one here is this other one is a very old drug. It's called Nabilon. And this went out as a real drug, and it's been used. It's called Sesament, and produced by Eli Lilly, and it's still used in Europe, mostly in vomiting. There's a resurgence of interest in this molecule. It's a very important compound. It has a lot of good properties. Now, in the nutraceutical field, we all know about, and probably the most interesting of all, cannabidiol, which is being honored today with this very well-deserved event, because it's really a wonderful finding in the cannabinoid field. So we have cannabidiol, we have THC. Then we have, if you look at this table, it has 5 carbons. If you count them there, if you cut 2 of them out, it gives you a compound called tetrahydrocannabiferine. Now this is, again, made with the same machinery, biochemical machinery of the body, but it has 2 carbons less in the table. It's much weaker. It has its own properties. It's being explored as we speak. Then if you take cannabidiol and you cut the 2 carbons, you get the cannabidiol variant. Okay, so now, so the plant produces THC and cannabidiol, etc. But what happens in the body? We found that there is some machinery in the body, and it wasn't made for THC, it must have been made for something else. So it did, in fact, have its analogous products, and they're called endocannabinoids. So we have then the phytocannabinoids, represented by THC, and we have the endocannabinoids represented by this molecule right here, called an amylide. On my initial presentation, I had a piece for this was getting in. Let's see if, oh, it does. Okay, great. So this is anandamide. Now, it obscured where it goes. It's a bit of a glitch switching systems here, computer systems. But this anandamide essentially, if you're having docs right here, this is where we did, and I showed exactly how it gets in. In fact, it's manufactured by the cell itself. The membrane itself has these ingredients, and I'll show you how they can manufacture this anandamide. Now, looking at this biochemical system, it's actually quite a complex system. However, you'll see two representatives that come out all the time. One of them, as I mentioned, is this one. It's anandamide or arachidoryl ethanolamine, and this is a glycerol 2-acylglycerol or 2-AG. And there they are. Actually, they're not very potent molecules. If you compare them to THC, they're less potent. In vivo, anandamide is maybe 3 or 5 times less potent than THC itself. Well, not only this, but in the body, there are these enzymes that deactivate them. They just chop them. They do esterase, it cuts the molecule right here, and these two pieces then have no activity at all. And here you see, this piece is called arachidonic acid, and this is glycerol, in this case it's called ethanolamine. It turns out these two guys have very different menus. They're involved in different activities, and sometimes they come together, they synergize, sometimes they don't. And there's a whole story. We don't have the full story about it. But it's a very intriguing story about when one takes over and which properties, which physiological properties are involved, they're involved in. The other thing to keep in mind is that these molecules are produced in different topographies in the body. Even within the brain, 2-AG and anandamide are produced in different areas. So you can have then selectivity based on that. Now, okay, so we have 2-AG and anandamide, simple system. It's actually not that simple. It's a whole bunch of these lipids that are around. It includes also lipids that are produced by the omega-3s and the omega-6s and so on. And there's a very big combination of all of those. And all of these are involved in the properties of the cannabis effect. And they're present in different amounts. We've developed a method where we can measure the profile of all of these. You can have a drop of blood, put it there in the mass spec, and you know exactly what it is. We can see, for example, that in certain pathological conditions, there's a disturbance of these, and we try to use these as metabolic biomarkers. So I won't go through all of these, but you'll see, you recognize that some of them have these omega-3s and omega-6s there. It's an example of the omega-6 here, example of an omega-3 right here. Okay, about these receptors. So what is the switch that produces these effects? There are two switches, two receptors, two GPCRs, which is G protein coupled receptors, and this is the CD1 receptor, and this is the CD2 receptor, and they have a lot of homology. Turns out that they do different jobs. This receptor, the CD2, which is a little smaller, is mostly involved in the immune system. And a lot of its activities in the immune system is present in the brain, but in very small quantities until there is something happen to the brain where you have an inflammatory effect or a neurodegenerative condition like Alzheimer or cancer. Then you get a big explosion of these CD2 receptors. So they're produced, they're upregulated upon demand. On the other hand, CD1 is present in a lot of places, and I'll show you where. And a lot of the THC effects that we know are related to the CD1 receptor. Where are they? They're everywhere. And you see here that in the brain you have mostly CD1, except of course when you get to inflammatory conditions. I mentioned Alzheimer. It could be other, for example, Huntington's disease, you get large amounts of the CB1, the CB2 expressed in the brain. So in the heart, you get CB1, and in the lungs you get both. Blood. We found it also in sperm. So human sperm has CB1 receptors, and it's thought to be involved in the interaction of the sperm where it gets anchored inside the egg. And by doing this, it affects the process of implantation of the egg, of the sperm in the egg. Now, this is complex. I'll just give you a little bit of tidbit on this, so as I said, this is a very special kind of a system. It's unlike the systems that we had known, like the dopamine system and the serotonin system, and a lot of you have known that there's a lot of drugs that came out of this system. The difference here is that these molecules are made from the membrane itself. They start from one piece like this, a bunch of enzymes come in here and produce it. So what you can do then is you can interfere with this, make compounds that interfere with this production and increase or decrease the levels of this molecule. So we have these enzymes here, and these other enzymes that make 2AG. Then we have two enzymes that are involved in breaking down, because you don't want to have a continuous production of these products and have uncontrolled concentration. So as soon as they do their job, and these two enzymes here, one of them is this enzyme here, which hydrolyzes one of the two molecules, and this is the other enzyme which hydrolyzes an anamide. So this is called fatty acid hydrolase, and this is called 2-acetylglycerol lipase. We worked extensively with these enzymes. There's good opportunities there. If you think about it, if you stop these enzymes, then you get an overproduction of the particular molecule, of the particular neurotransmitter there. So one way of producing therapeutic effects is to block these enzymes. And we know now, for example, that in pathological conditions, there's an overproduction of these molecules. And learning from this, then, we can block these and enhance the natural propensity of the body to protect itself against change. Okay, so natural propensity is to have a homeostatic equilibrium here. You go beyond that, then the body protects itself by making these, and if we block that enzyme, we increase that protection level. And it turns out that these compounds like these, for that, would work well for conditions like neurodegenerative conditions. Again, here just to show you where these enzymes work. You see, this is one of the two enzymes that hydrolyzes the 2AG. This is the enzyme that hydrolyzes anandamide. And then, what I said before, you can block this here, increase the levels of this molecule. Now, let me talk about cannabidiol. Cannabinoid is the prince now of this situation, and I'll show you some information on it. We have this oral availability. It can be inhaled. It's excreted either as an oxidation product or in glucuronide. And here it is. If you eat, if you ingest cannabidiol, some of it will go to the liver and then subjected to oxidative deprivation here. Makes the acid, this cannabinoid acid, cannabidiol acid, or it could go into the urine and in the kidney and gets again glucuronidated and so on. So one reason these things have a long time of action is because they get stored with a lipid mechanism and then slowly they get released. Therapeutic effects, you'll hear more about them later in this presentation. And I just wanted to finish by touching on the mechanism with which cannabinoid works. We don't know the full story of it, but there are three, and it's not a single mechanism, but there are three mechanisms. So CBD works through the cannabinoid receptors, it's one mechanism. It also works on something called the vanilloid receptors, and the vanilloid receptors are the ones that are activated by chili peppers. And it also works as an antioxidant. So we summarize this here. So this is the substance that is in chili peppers. It works on this VR1 receptor. This is the endocannabinoid receptor. And then as an antioxidant. So we need to talk about these radical oxidative species here, reactive oxygen species that produce very nasty stuff called hydroxy radicals that go and chew up a lot of the lipids and produce bad effects. That's why these omega-3s work well, etc. and they're subject to it. So cannabidiol works on this, and I'll show you how we do. So here we are. In a natural situation, there's a balance between the antioxidants, because the body has its own antioxidants. And then the reactive oxygen species come from outside. As I mentioned, as I showed in the slide, they're coming from different stimuli, light, anxiety, all sorts of things. So if we have a balance between the two, then we have an equilibrium. However, when the equilibrium is disturbed, then we have a problem. So this is where, by giving cannabidiol, we reduce the reactive oxygen species. Alternatively, the oxidative, the antioxidant compounds in the body may have diminished. We give them cannabidiol and we have. So this is pretty much the story. I'll stop here and I thank you.
We're going to have a 15-minute break, and Dr. Makriyannis will be right over here, and you can ask him some questions. I just wanted everybody to just breathe for a second. Did we all notice how many cameras were taking pictures of the beautiful, elegant, complex slides on the screen? Did everybody notice that? That is amazing, isn't it? Because part of what we believe this change is all about is us getting serious about the science. This industry has done a very, very poor job of insisting rigor, haven't we? So if it gets quiet, and you have to listen very closely, and then you have to read and study, I understand that we all want simplified messages for marketing, but we don't want oversimplified messaging. And more importantly, the younger audience that are technological natives, that have grown up on their phones, that are fact-checking everything, they are insisting this kind of rigor. Let's celebrate that with Dr. Makriyannis.